So we're going to start here with that final character in the list of lymphoid tissues. That's the thymus. The function of the thymus is to positively and negatively select T lymphocytes. T lymphocytes, like all cells of the immune system, are born as hematopoietic stem cells in the red bone marrow. But even in utero, as early as in utero, these T lymphocytes, called naive T lymphocytes, are going to leave the red bone marrow, get out into the blood vascular compartment, and find their way to the specialized structure called the thymus. And in the thymus, there's going to be this conversation between these naive T lymphocytes and a series of epithelial cells found in the thymus itself. It's called stromal epithelial cells. And that conversation, and I'll anthropomorphize it a lot, and the conversation basically is selecting for T lymphocytes at two levels. It's first selecting for T lymphocytes that can recognize protein. If a T lymphocyte can't recognize protein, it's of no use. And so that's the first selection. Then after that T lymphocyte is selected to be able to find and bind to proteins, then the next round of selection is what kinds of proteins do you bind to? If you bind to proteins that are part of our own system, if you recognize proteins on our own cells, then you're dangerous. So you can't let those naive T lymphocytes mature into killer T cells that will harm our own cells. So they have to be eliminated. And that's what's going on here in the thymus, and we'll talk about that. I'm going to simplify it a whole bunch. It's really, really well understood. If you want to, you can read some reviews on it, and they talk about all the levels of control and how there are different transcription factors and different genes turned on and turned off, and depending on what proteins are produced when, that's all part of the selection process. I'm going to just talk about how stromal cells say, okay, you're good or you're not good, and we'll let it go at that. Then we're going to see that when we think about the immune system, we can divide the immune system into these two great categories: an innate or nonspecific immune system and an adaptive or specific immune system. When we think about the innate or nonspecific immune system, we're really talking about a very rapid response. This is the first line of defense against potential pathogens. This response comes up in a matter of hours. It's very quick. But it's not highly targeted. It's pretty capricious about what it destroys, and it does a lot of damage. And so we'll see that in this innate immune response, we just get a lot of havoc produced. But in that havoc, in that chaos, what we're doing is we're mounting a short-term holding action against those pathogens. And we're mounting a short-term holding action until our adaptive or specific immune response can gear up, engage, and produce a particular response to that particular pathogen. When we think about the cells of innate immunity, we think mostly about granulocytes. And again, this is a cell type you find in the blood. Many of you may have seen these if you look at your own blood panel after going to the doctor. And the granulocytes come as neutrophils, eosinophils, and basophils. The granulocytes originally got their name in the 19th century because when the 19th century histologists looked at these cells, then what they saw is they saw these cells chock full of granules. Their resolution wasn't good enough at the time to see vesicles, so they just said they were granules because they're big. We now know that these are basically just great big vesicles. They divided them into neutrophils, eosinophils, and basophils simply upon the kinds of dyes that would stain them. Neutrophils stained with neutral dyes, eosinophils stained with this particular dye called eosin, and basophils stained with basic dyes which were at a basic pH, alkaline dyes. And that separated these three out. We'll talk about them briefly. Most of our time is going to be spent on the neutrophils because they're going to play an important role in the non-specific response to bacteria. We're going to divide the innate response into two big categories, again, glossing over the surface. One of them is going to be interferon. Interferon is going to be the response to viruses. So we'll couch this in terms of the infection of the SARS-CoV-2 virus of our epithelial cells and how that happens and how our cells begin to respond to that. And that first response, the initial response, the innate response is going to be through interferon. then hopefully if the interferon can hold that SARS-CoV-2 virus at bay long enough, then it won't progress to a more serious disease, and your specific immune response can engage and throw off the infection. The other innate response we're going to talk about is with bacteria, and this is inflammation. The signs of inflammation have been known for close to 2,000 years. They were first described by a physician called Galen, and Galen was a physician in Anatolia, and he was a physician for the gladiators on the Anatolian Peninsula. So he got plenty of chance to see inflammation in wounds. And so he generated some general ideas about inflammation, and we still use the so-called cardinal signs of inflammation that he developed 2,000 years ago, and we'll talk a little bit about that. And we'll also see that in the adaptive immune response, so the first story is going to be about the innate immune response, then we'll talk about the adaptive immune response. The adaptive immune response has these characteristics. It's slow. Its initial response takes days to weeks. It's highly targeted, where the innate immune response is pretty capricious. It blasts away at everything. The adaptive immune response is going to target 16 to 20 amino acid sequences on the foreign or pathogenic protein. So it's highly targeted. The other thing is, unlike the innate immune response, which every time you get infected, it does the same thing over and over again, the adaptive immune response learns, and it has memory. So that if you're exposed to a pathogen once, you should be able to mount an immune response sometime later. Again, this varies, as we're seeing with SARS. It looks like these vaccinations that we're getting do wane over a period of months. And so the memory isn't perfect. Then there are branches of the adaptive immune response. I want you to hold on to these ideas. The branches of the adaptive immune response are the humoral branch and the cellular branch. The humoral branch is carried out by B lymphocytes. When you see humoral branch, associate the humoral branch with the humors, including blood and plasma. This is the old humoral days of phlegm and bile and such, but we can think about it as fluids, as being plasma and being extracellular fluids. These B lymphocytes are going to be secreting whole protein antibodies out into the humors, out into the extracellular space. So that's the humoral branch. The cellular branch uses these cells, the T lymphocytes. And not today, but on Monday, we'll see that the way the T lymphocytes work is that they can only recognize foreign proteins by cell-cell contact. These guys just dump tons of antibody out into the extracellular environment, and those antibodies bind to antigen. Here, the response is a little more subtle. Here in the T cell response, the T cells themselves produce something called a T cell receptor. That T cell receptor can only recognize foreign proteins that are expressed on other cells on particular proteins. We'll see MHC1 and MHC2 are going to be the display proteins that will be recognized by the T cell receptors on these T lymphocytes. But most of this story is going to come on Monday. But let's start out with positive and negative selection of T lymphocytes in the thymus. We mentioned this yesterday. In utero and shortly after birth, our thymus is this big structure. But by the time we reach puberty, it's pretty much atrophied. But it plays a really important role in our immune system, and it starts out, even in utero, in the red bone marrow, where we have hematopoietic stem cells. Among those hematopoietic stem cells will be the cells that produce T lymphocytes. Now, when these T lymphocytes leave the red bone marrow, they're said to be naive T lymphocytes. These naive T lymphocytes must, before they can be functional, they have to pass through the thymus. So I'm going to draw this box here and label it as a thymus. So this is intended to be the thymus. And in the thymus, there are all of these epithelial cells. These are called stromal epithelial cells. And it's these stromal epithelial cells that do the job of T cell selection. And this T cell selection is first for efficacy. That means, are these T cells effective? And second, for specificity. And that's going to be, what kinds of proteins can they see? That's where we're headed with this. So we can imagine one of these naive T lymphocytes coming out of the red bone marrow, finding its way to the epithelial cells in the thymus. Now, if you get deeper into this, you're going to find these stromal epithelial cells are in different regions. We do different selections in the cortical region, the medullary region, but for our purposes, this T cell, this naive T cell, binds to the stromal epithelial cell. And there's a kind of welcome. Hello, how are you? And the epithelial cell says to the T cell, can you recognize proteins? Do you have the ability to recognize proteins that are expressed on other cells? And if the answer coming from this naive T cell to the stromal epithelial cell is, no, I can't really recognize proteins on other cells, then this epithelial cell says to this naive T cell, you're useless, go die. In fact, what's happening is this epithelial cell is now withholding growth factors to go to this T lymphocyte. And if it withholds growth factors to the T lymphocyte, then we turn on the apoptotic machinery and the cell dies. But the trigger for this is that this cell is incapable of recognizing proteins on other cells, so it's not going to be a useful T cell. Because as we said in the introduction, T cells have to recognize, to be effective, they have to be recognizing for proteins on other cells. But then this naive T cell, another one comes along. We have another T cell here. Comes along. Same process. Handshake, hello, how are you? The epithelial cell says to the T lymphocyte, can you recognize proteins expressed in other cells? And the naive T lymphocyte says, sure, I can do that, no problem. And it says, good, you passed the test. And it passes this naive T cell to the next epithelial cell. And the epithelial says, well, congratulations, you passed the first test. Now I'm going to ask another test. I'm going to ask another question. And the epithelial cell asks the T lymphocyte, it says, what kinds of proteins can you recognize? And if this naive T lymphocyte says, I don't care, I'll recognize any protein on any old cell, then this epithelial cell says, you're dangerous, and withholds growth factors, and the cell dies. The reason that's dangerous is that if this cell is producing proteins that can recognize proteins on any old cell, any cell in our body, then this could potentially become a T killer cell and kill those cells. That's an autoimmune disease. So you want to be pretty careful when you're selecting these T cells, first that they're effective, that is positive selection, which says the T cell recognizes proteins. It's necessary for this T cell to recognize proteins, but it's not sufficient. Because if this T cell is promiscuous and recognizes proteins helter-skelter all over the place, then it's dangerous. So in fact, what's going to happen here is this T cell is going to be handed from one cell to the next, and each one of those cells is going to express a family of proteins. And if at any point this naive T cell recognizes one of those families of proteins, then growth factors are withdrawn and this cell dies. That's the way we select for efficacy and specificity, so that T cells, or T lymphocytes, recognize only foreign proteins. And don't recognize our own proteins. This is what happens in a number of autoimmune diseases. Our T killer cells go out and they begin to recognize proteins in our own cells as foreign, and they begin to kill those cells. Classic example of that is type 1 diabetes. Type 1 diabetes, our immune system begins to recognize the beta cells in our pancreas that are secreting insulin and kills those cells. And from then on, those individuals have to have daily injections of insulin because their immune system screwed up and killed those cells. And there are any number of diseases growing by the day that we find as autoimmune diseases, that where the immune system has made a mistake and it's killing our own cells. So any questions on that? Yes?
What is the negative selection come into play?
So this is the positive selection. Then each time the naive T cell moves down the road and it's tested, can you see proteins? The negative selection is that you come to the end of the line and it has not recognized any proteins that are the self-proteins that are expressed on these cells. So along this sort of conveyor belt, along this series of way stations, each of these epithelial cells will express a different self-protein. And at each way station, there's the question of whether or not the T lymphocyte can recognize those proteins. And so to become an effective T lymphocyte, it has to go through all of these stages and come to the end and never on any of those cells recognized one of those proteins, and that's negative selection. It was unable to recognize the protein. Yes?
So is it in an autoimmune disorder that your stromal epithelial cells, like they're the ones not working because they're not checking things that don't recognize?
In some cases, we think it's a stromal epithelial cell problem. In other cases, it's more subtle. In other cases, we think that what's happening is we'll get a viral infection, and we don't throw it off soon enough, and then we continually have these viral proteins out on the cell surface. And then the immune system, over time, makes a mistake and there's a good enough match between the T cell receptor on the T killer cell and that viral protein, and says, I guess that's good enough, and it kills the cell. So, and those two are possible, yes. Yes?
How does a test work? Like, how do they test each cells?
How do they test?
Protein recognition, specificity.
My understanding of this test is it's a protein-protein interaction. That is, there's going to be a protein which is going to be, I think, there'll be a protein expressed here on the surface. And then this cell is going to produce a protein which is one of our own proteins. And if this, usually an MHC1 or MHC2 protein, on this T lymphocyte recognizes one of these proteins, then that's dangerous. So it's got to go through the whole series, and you have to come out the other side. And this cell, in this paradox, it must fail to recognize any proteins along the way, except for the first.
Also, it has to recognize the first one.
The first one is, are you competent?
Oh, are you able to?
Are you able to? And then the next one is, okay, you're competent, you're able, but don't be too able.
Okay.
And so then it has to be passed from hand to hand to make sure that it never recognizes any of the proteins that are self-proteins. Anything else? Okay. So that's basically how we select those T lymphocytes. So now let's talk a little bit about the immune system. And we can organize the immune system at different levels. So we're going to take this heading and call it the immune system. The immune system now is going to be divided into these two categories. One is said to be the innate, sometimes known as the non-specific immune system. And the other branch here is going to be the so-called adaptive or specific immune system. This adaptive immune system is going to be further subdivided into a humoral branch. The humoral branch is characterized by B lymphocytes that secrete antibodies. They're secreting antibodies into the humors, and they're into the blood, into the serum, into the extracellular space. So these are whole antibodies being secreted, just dumped out. The other branch here is said to be the cellular branch. And the cellular branch, too, is divided into two categories, one of which is called the T helper cells, and the other is called T killer cells. Whether we're dealing with T helper cells or T killer cells, in this case, these cells recognize foreign proteins. They recognize foreign proteins displayed on other cells. They recognize these foreign proteins displayed on other cells using a protein called a T cell receptor. So the T cell receptor, we'll see on Monday, is the transmembrane protein that recognizes foreign proteins on other cells. Those foreign proteins we'll find are either expressed on MHC1 proteins or MHC2 proteins, but let's hold that for Monday. These guys now are secreting antibodies into the extracellular space. These guys require cell-cell contact. So keep that in your head until Monday when we get down into the weeds of that. The innate or non-specific branch can now also, for our simple purposes here, be divided into response to viruses and the response to the viruses is interferon. There will also be a response to bacteria. In this response to bacteria, we're going to subsume under inflammation. But let's begin here with the interferon. And let's tell the story of the interferon from the point of view of something that's going on all around us these days. So we'll tell this story from the point of view of epithelial cells becoming infected by the SARS-CoV-2 virus. The SARS-CoV-2 virus is a coronavirus. It gets its name as a coronavirus because it has these spikes sticking out all over it. And when people first recognized it, those spikes looked like a crown to them, and so they named this family of viruses the coronaviruses. And this is one particular kind of coronavirus that we're dealing with right now. It looks like it came out of a bat population someplace in China. It looks like it's endemic in the bat population. But once it's infecting, but now we have receptors for those coronaviruses on our cells that allow the coronavirus to infect our cells and then proliferate in our cells. And these coronaviruses are binding to epithelial cells in the respiratory tract. And that's why this now is a respiratory disease. In extreme cases, it causes pneumonia and death. Well, let's start out with the idea of SARS-CoV-2. Let's label this as interferon. And we'll start out with the virus, the SARS-CoV-2 virus. This, of course, causes the disease COVID-19. The way it begins is that we can imagine this sort of row of epithelial cells. And these are the respiratory epithelial cells. The respiratory epithelial cells are those cells, the epithelium that lines your nose and your throat all the way down into your lungs. And these respiratory epithelial cells have on their surface a transmembrane protein. All of them have this transmembrane protein. This transmembrane protein has a name. It's called ACE2. We're going to run into a relative of this protein later on when we talk about the kidney. And this is called angiotensin-converting enzyme. And the angiotensin-converting enzyme is this enzyme that's on endothelial cells and regulates blood pressure. The ACE2 is a related gene, but it's different. Where ACE1 is mostly on endothelial cells, ACE2 is on epithelial cells. The ACE2 now is on all of your epithelial cells. And now here comes the villain in the story. This is the coronavirus. It has these spikes coming out of it. It has a proper phospholipid bilayer. Across the phospholipid bilayer is the spike protein. Inside the phospholipid bilayer is RNA because it's an RNA virus. But for our story today, what's going to happen is this spike protein is going to be able to bind to the ACE2 receptor. So we can think about this ACE2 protein as being sticky for the spike protein. So now this virus is bound to the cell through the ACE2 receptor. Now there's a bit of controversy. In some quarters, it's thought that this ACE2, along with the virus, has to be endocytosed and go through an endosome, and that's where most of the rest of the story occurs, where we're going to allow it to get into the cell. My understanding is that the most recent data say that particularly with the most recent of the variants, the Omicron and the Delta variants, that this protein now can be broken, cleaved on the surface of the cell. So the next character in the play is going to be this transmembrane protein, and I'll put it in red. This protein has a name. It's called TMPRSS2. It stands for transmembrane protein serine site 2. This is a protease. For the biochemists among you, this is a serine active site protease. So in the active site, it has a serine, but it's a protease, and what it does is this protease cleaves the spike protein. If we look at one of these spike proteins coming out of as a transmembrane protein, there's a region right here called the S1, S2 interface. Turns out there are a bunch of arginines in here. That's not important. But it's the arginines probably that are a signal so that these TMPRSS2 can bind this thing and they cleave it. It's a protease. The spike protein is a protein. So you cut the protein, and when you cut it at a particular spot. Now it turns out that on the spike protein, toward the virus side, there are a series of amino acids. And these are predominantly hydrophobic amino acids. Now again, up until a few months ago, we thought that the process was it would bind, be brought into the cell by endocytosis. Then this TMPRSS2 protease activity would happen in the endosome, in the vesicle, and then it would escape across the vesicle. But more recent data suggests that when we clip this here, this series of hydrophobic amino acids now allows the virus to come right down onto a neighboring cell. When it lands on the neighboring cell with this series of hydrophobic amino acids exposed, that allows it to insinuate itself into the phospholipid bilayer of the cell. And one way or another, that now permits the access of the coronavirus into the cell. The virus uncoats, and the RNA, which is the genetic material of this virus, takes over and begins to make viral proteins. When this virus invades this cell, the viral RNA co-opts the protein synthesis machinery of the infected cell to make viral proteins. It's a remarkably simple structure. It only has about 13 genes. It makes more proteins than that. It has some clever tricks to make more proteins. But it's dangerous because once it invades the cell, this cell no longer can make its own proteins. It's now destined to make only viral proteins. Well, the first line of defense here is that this virus-infected cell, the virus-infected cell, in response to the viruses, produces and secretes interferon. The interferon response is not particularly specific. What the interferon does is it now becomes a signal to neighboring cells. This virus-infected cell's a goner. There's nothing we can do about it. It's done. The interferon now binds to receptors on neighboring cells to slow down or stop endocytosis, exocytosis, and protein synthesis. But if you think about it, that's a pretty drastic thing to do. This virus-infected cell is telling all of its neighbors, slow down your metabolism. Stop taking things in by endocytosis. Well, that's useful because it's not going to endocytose these viruses. It stops exocytosis. That's particularly useful rather, because these viruses escape by exocytosis. The viral machinery is taken over the cells to make its own proteins, and so by suppressing protein synthesis, you're now reducing the rate of virus production. So that's what the interferon is doing. It's a non-specific, it's an innate response to tell everybody in the neighborhood, slow things down so we can slow down the replication of the viruses so that we can generate a specific response in a couple of weeks. So this is all a holding action. Now, if you read more details about these viruses, even though they only contain about 13 genes, and they only make about, I think it's 15 or 18 different proteins, some of these proteins, when they take over the cell, are dedicated to controlling the whole cell mechanism. They go out and tell the cell not to make self-proteins anymore. They tell the cell, we're only going to make viral proteins. They gather together the endoplasmic reticulum so that they can use the endoplasmic reticulum to make the viral structures. They basically take over the cell. My vision of these proteins is it's a little bit like the Peaky Blinders story where the group takes over one of the competing gambling operations, and Arthur arrives and says, we're in control now, you can't do anything for yourselves. That's what these viruses are doing. They're telling the cell, you can't do anything for yourself now, we're going to take over. And they then control protein synthesis. And the job of the interferon is to slow down that process, keep it from spreading from cell to cell, until your specific immune response can mount a decent specific response and throw off these viruses by making antibodies to basically prevent them from binding to your cells. So questions on that? Yeah?
Does the interferon occupy the H2 receptor, or does it have its own?
The interferon has its own set of receptors. And I've seen it referred to as a G protein coupled receptor, that it starts that cascade of events, probably inhibiting all manner of enzymatic activities. But it's a separate receptor. And I should point out that you don't want to be releasing the interferon in the apical side, right? This is going to go into your throat and gone to your nose. You want to release the interferon on the basilar side so it can bind to receptors on neighboring cells. Anything else? Yes?
So the point of infection isn't the spike protein binding to ACE2, it's after it's chopped and then hits the cell next to it?
So the initiation of the infection, I'm not sure where you want to put the initiation. It first has to bind to the ACE2 protein on the cell. That's the first step. But that's not enough. One way or another, you've got to clip that spike protein to expose those hydrophobic amino acids to give that virus access into the cell. And again, since you've asked the question, if we blow this up, I've got to do it back here. This is the other model, and this is the model that prevailed for a long time and makes more sense to me. So in this case, the ACE2 binds to the virus. This whole complex then is taken into a vesicle. And we now have this in a vesicle, or probably an endosome, where the ACE2 is bound to the virus. And it's within the vesicle, or in this endosome, that we have now the activity of this protein, which is the TMPRSS2 protein. And under this model, what will happen is we clip the virus loose so we cut the virus at this point. We now have this portion of the spike protein that's hydrophobic in the endosome or in the vesicle. It now fuses, binds with the phospholipid bilayer, and gets out from the endosome into the cytoplasm. This is the model that they originally had. But my understanding is that model is changing with the most recent variants, and they're thinking that one of the reasons that the Omicron and Delta variants are so much more transmissible is they don't have to go through this process. They basically get clipped on the surface, and then they can infect all manner of other cells in the neighborhood. That's the model. But these things have been changing for the last 2 years, so who knows what we'll know in the next 6 months. Yeah.
So, is it possible for these cells to survive this, or are they just done?
Once the virus has, once it uncoats and its RNA begins to make its own proteins, those proteins now are going to overwhelm the cell. It's a goner. It's dead. There's nothing it can do. It's not recoverable. And so that's why, since this is such an extreme event, that the cell's going to die, that's why we want to prevent neighboring cells from getting infected. So that's the effect of the interferon. And our immune system has not seen this thing before. So this is the first line of defense. To make things even worse, among those proteins that are produced from the viral proteins, one of the first things those proteins do is they suppress the interferon production. Of course. Yeah. So any other questions? Okay. So that's the story here for the interferon. So now let's begin talking about this story, which is the story of the response to the bacteria. This is one we're all familiar with. We all know about this one. We've all experienced this. We've all, at one point or another in our lives, driven a splinter under our skin, and we say it becomes infected. And it hurts, and it turns red, and it swells, and it exemplifies all of the so-called cardinal rules of inflammation. So let's start here and think about what these bacteria delivered under the skin are doing to start the process of inflammation. And again, this is one form of inflammation. If you get into the literature on inflammation, there are a whole bunch of different types of inflammation, but this is one particular type. And so let's label this as inflammatory response. To bacteria. So let's imagine we have a stratified squamous epithelium, which we know is skin. It's the epithelium of skin. It sits on basal lamina. And beneath that basal lamina is, of course, the dermis. This red block here is going to be the dermis. And running through the dermis, of course, will be capillaries. And the capillaries are lined with an endothelium. Then out here, at the interface between the epithelium and the dermis, are going to be another set of cells called mast cells. And the function of the mast cell is to secrete histamine. Now we can imagine that we get a splinter. We take one of these splinters and we deliver it. We basically use it to drive down underneath the epidermis. And it's carrying with it a load of bacteria. These are meant to be bacteria. And this load of bacteria now initiates a response, and we'll go through the cascade. But first I want to list here the cardinal signs of inflammation. The cardinal signs of inflammation are first redness, heat or warmth. Sorry. The second one is swelling. The third one is heat or warmth. And the fourth one is pain. As I said in the introduction, these cardinal signs of inflammation were recognized 2,000 years ago. And they're easily recognized because every time we get a splinter or we cut ourselves, we get a bit of an infection, we see these signs. It turns red, often swells. It gets warm because increased blood flow, and it hurts. To this day, these signs of inflammation in medical schools are taught by their old Latin names, which are rubor, tumor, calor, and dolor. It's a nice ring, rubor, tumor, calor, dolor. It's easy to remember. So rubor is redness, swelling is tumor, heat is calor, caloric, pain is dolor. And then after we go through these series of redness, swelling, heat and pain, rubor, tumor, calor, dolor, we enter another phase. This is a phase where we say, I feel terrible, I'm gonna die, it's awful. And that phase is called Eeyore. That's a joke. But nonetheless, we have these cardinal signs. Let's come back here and see if we can fit those cardinal signs into what's happening with the bacteria. The bacteria now are generating, these bacteria are maybe killing cells. And the bacterial proteins these bacterial proteins activate the mast cells, and the mast cells are said to degranulate. These mast cells are chock full of these granules, which are vesicles that contain histamine, so they release or secrete the histamine. One of the things that the histamine does is it will bind to smooth muscle. It will bind to smooth muscle on the arterioles. Now, we talked about the alpha-1 receptors causing that smooth muscle to constrict. Well, the mast cells cause smooth muscle relaxation. Well, you'll know that when we relax this smooth muscle, we're now going to get increased blood flow through this region. So the bacteria have told the mast cells, release histamine, we increase the blood flow, and that increased blood flow can account for warmth and redness. Swelling. Well, another thing that the histamine does is it affects the endothelial cells. It affects the endothelial cell tight junctions. So it actually causes these tight junctions to let go a little bit. So by releasing the tight junctions on the endothelial cells, you make bigger openings. You now have more flow through this area, and there's less resistance to the movement of fluid from the blood vascular compartment out into the extracellular space, so now we get swelling. So we've accounted for these three. Pain actually is on its own. These toxins produced by the bacteria will actually open receptors here in the pain neurons, the so-called vanillinoid receptors, and that will actually cause pain. In the final minute, let me say one of the other things that happens here is that there's a signal. There's a signal that tells these endothelial cells there's a signal to place selectins on endothelial cells. These selectins are now going to bind to neutrophils. And we'll finish the story on Monday by showing that these signals coming from the damaged cells and from the bacteria are going to wend their way to the red bone marrow. And the red bone marrow, we're going to mobilize neutrophils, which are now going to stick to these endothelial cells, leave the blood vascular compartment, and come out into the site of bacterial proliferation to attack the bacteria. We'll begin that story on Monday.